Thank you, Dr. Radhika, Dr. Kumar, and Dr. Namrata, and Dr. Vanati. So the NEI Audacious Goals Initiative, the NEI Restoring Vision through Regeneration of the Retina. The Audacious Goals Initiative is an effort by the National Eye Institute to push the boundaries of vision science by facilitating cross-disciplinary research. The AGI is tackling the most devastating and difficult to treat eye diseases. So, interferometric optophysiology of the human retina, Dr. Ruda and colleagues are designing a system to map the interaction of the cells in the retina. The system will enable scientists to stimulate individual neurons and observe other cells as they become active and response. And mapping these intricate signaling patterns will help explain how the retina processes visual information before it is sent to the brain, and will be an important tool for monitoring function in regenerated cells. The system will incorporate eye tracking components and adaptive optics, a technology that corrects for distortion imposed by the cornea and the lens. And accelerating vision restoration with in vivo cellular imaging of retinal function. Dr. Williams' team is designing an optical system to image responses to light of large numbers of individual cells in the retina. The system uses two main components, a fluorescent marker that can detect cellular calcium fluxes, and two-photon microscopy, which uses infrared light to detect the fluorescent signals without damaging living tissue. The team plans to test their system in collaboration with the investigators who are exploring 3 different approaches to vision restoration: preserving photoreceptors with gene therapy, replacing lost photoreceptors using stem cells, and genetically re-engineering cells other than photoreceptors to respond to light. A two-photon ophthalmoscope for human retinal imaging and functional testing. Dr. Palexevsky and colleagues are pursuing a tool to visually monitor vitamin A derivatives in the retina, and vitamin A derivatives help power the light-sensitive machinery inside photoreceptors. Many inherited diseases of the retina involve mutations that affect the retina's ability to utilize or recycle vitamin A, and Dr. Palexevsky's team will develop a two-photon microscope capable of measuring the metabolism and distribution of vitamin A derivatives within photoreceptors at baseline in various retinal diseases and in response to potential therapies. Imaging optic nerve function and pathology, Drs. Song and Wang are adapting two technologies, diffusion-based spectrum imaging and diffusion functional magnetic resonance imaging, to non-invasively visualize the optic nerve. And although this bundle of fibers originates in the retina, most of the optic nerve resides deep within the brain, out of reach of most devices used to see into the eye. An optic nerve damage, a consequence of glaucoma and other optic neuropathies, is currently irreversible. And this system could be used to monitor how a patient's optic nerve responds to a potential new therapy throughout the course of treatment and without the need for biopsy. Platform technologies for microscopic retinal imaging, development and translation, with collaborators at several research institutions. Drs. Dubra and Carroll will develop a suite of core technologies that will advance and increase the usability of next generation retinal cameras. The suite will include real-time eye motion stabilization, image resolution doubling, a tunable lens to improve the focusing of all colors of light, and high throughput methods for testing the function of individual cells. And about the initiative, the NEI, the Audacious Goal Initiative, is catalyzing research that will enable the restoration of vision through regeneration of the retina. And the central goal is to replace cells of retina that have been damaged by disease or injury, and to restore the connections to the visual centers of the brain. And through strategic research funding, the NEI is enlisting dynamic scientists and teams who are developing the necessary knowledge and technology to make the goal a reality. They have a lot of funds, but only used for this particular area, and that's what Paul Seeving presented in the RO last year. The AGI began with the Audacious Goals Challenge, a prize competition that challenged participants to imagine the greatest achievement for vision research during the next 10 to 15 years. The challenge attracted more than 450 innovative proposals from around the world, and the NEI consolidated the proposals into 6 themes, which were further explored by leading scientists at the Audacious Goals Development Meeting. In consultation with the National Advisory Eye Council, the NEI chose to pursue the goal of restoring vision through the regeneration of neurons and neural connections in the eye and the visual system, specifically targeting the photoreceptors and retinal ganglion cells. The AGI Steering Committee plots the scientific trajectory of the AGI, consisting of 3 to 5 leading researchers. The steering committee is responsible for identifying knowledge gaps and the scientific expertise needed to bridge them. The National Advisory Eye Council provides administrative guidance to the NEI, and the NAEC consists of 12 recognized leaders in the field of ophthalmology, optometry, and basic sciences, and the NAEC advises the NEI Director on AGI funding plans and provides a second level of review on proposal to AGI funding opportunities. The AGI office at NEI, led by Stephen Becker, provides logistical support for all AGI-related activities. The AGI working group comprises NEI staff within the division of extramural research who help draft funding announcements, monitor the progress of research programs, and make recommendations of the AGI steering committee, the NAEC, and the NEI director. What will it take to reach the goal? The AGI is charting new territory. Need to gain a better understanding of how retinal neurons mature, integrate with other cells, and extend their connections to appropriate regions of the brain. And need to develop better, more efficient technologies for producing authentic replacement cells that are safe for transplantation. Need new technologies to measure the function and growth of cells in the retina, and need to develop systems for deploying new knowledge and technologies in the clinic. And when will a NEI reach the goal? With adequate funding, the NEI projects reaching the goal in 10 to 15 years. NEI and its partners creating a new global vision for the research. NEI international consultants and collaborators across the globe are creating a common platform for eye research. With the lessons learned from AEGC, similar programs will be planned for other regions of the world, creating a global eye genetics consortium. Thank you.